So now that we've looked at the first and second lines of defense against pathogens by the body, we're going to dive into the third line of defense. Now, the third line of defense we're going to break down into two different responses. So in this lesson we'll focus on the humoral response, and in the other lesson we'll focus on the cell-mediated response. So the immune system and the immune response. So the immune system is all of the organs and cells that help to protect us against foreign organisms or pathogens. And the immune response itself is more the homeostatic mechanisms. So the stages in place to help prevent us from getting sick, to protect our internal environment to ensure it can operate normally. So as I mentioned, today we're going to focus on how antibodies mediate that response, and this is known as humoral immunity. So when we're talking about antibodies, we need to talk about where they come from. And in order to do that, we need to look at these particular lymphocytes, which again are a type of leukocyte or white blood cell. And these lymphocytes can be divided into two main categories. First of all, we have our B cells, and these B cells are produced in the bone marrow, and they also mature in that bone marrow. What they're going to do is they're going to defend against any extracellular bacteria, any extracellular toxins and viruses as well. And they are involved in our antibody-mediated immunity, or in our humoral immunity. Now, something that we'll look at in more detail in the next lesson is the action of T lymphocytes. Okay? Now, these T lymphocytes, the key distinction here is that instead of being produced and maturing in the bone marrow, they're produced in the bone marrow, but they mature in the thymus gland. These T cells are largely going to target intracellular pathogens, in addition to things like transplanted tissue and cancer cells, and this is what we call the cell-mediated immune response. So, the thymus gland. Okay, the thymus gland, as we've just said, is where T cells go to mature. So they come from the bone marrow and then they go to develop in this thymus. And what they're doing in this time is they're educated, okay? And they're educated regarding what the body's own proteins look like so that they can recognize cells versus any antigens that they pick up from somewhere else. So T cells then migrate from this thymus gland and they move towards the lymph nodes and the lymph nodules around the body. So in this diagram here, we've got a very quick representation of it. I'm just going to skip it for now, so feel free to pause and have a look at this one in more detail. As we move on down, when we're talking about the transmission of these particular particles around the body, it's important that we define these things really clearly. So first of all, our keyword here is antigen. We've spoken about these words before. Antigens are anything that is capable of causing an immune response. So previously when we've spoken about an antigen, it's been a protein carbohydrate marker on the surface of a cell. And in this instance, we're just looking at antigens as the marker on a substance capable of causing an immune response. Antibodies are our next key item. Again, we looked at these in year 11 when we're talking about different blood types, and these ones are Y-shaped proteins that are produced in response to non-self antigens, or antigens that come from outside our own body. Finally, our final keyword here is antigen-antibody complex. This is where we have an antigen and an antibody which are complementary to one another and they combine. So it's really important that you understand these three terms before we move on with any of the content. So, two types of antigens that we can find. We have our self-antigens, and these are found on all body cells. And these ones, generally speaking, unless we're talking about autoimmune conditions, won't cause an immune response. And secondly, we do have our non-self antigens. And these ones, the key idea here is that they are foreign compounds that will trigger an immune response. So as we move down, we're now going to tie our third line of defense into our second line of defense. And so in our second line of defense, we spoke about the process of phagocytosis by macrophages. And what we're looking at here is redefining what these cells do. So previously, we've mentioned that macrophages, as I'll write here, undergo a process known as phagocytosis. And when they do this, these macrophages themselves become, phagocytosis, these macrophages become an antigen-presenting cell, or an APC. So how do they do this? Well, number one, we have the phagocytosis of the non-self antigen. And during this process, we have the fusion of lysosome and vesicle together. And then the digestive enzymes from the lysosome will act on that pathogen and break it down. Breaking it down into really small fragments. And previously we've spoken about the exocytosis, so step 6 over here on this diagram, of these pathogen particles from the cell. But what we're really looking at with an antigen-presenting cell is step 5 instead, and so I'll highlight this in a different colour, where some of the fragments are taken and placed on the cell surface. And it's at this stage, once these antigens have been placed on the cell surface, that the macrophage itself has become an antigen-presenting cell, an APC. So in your exam, please make sure that you write antigen-presenting cell in full first, and then you can use brackets following that to write APC. You're going to need to do this for each and every question. So a very quick breakdown here. When we're looking at B cells, this is now coming back to our idea of this humoral response. So up the top, we have our B cells in our lymphoid tissue, so in the lymph tissue, lymph nodes around the body. And what happens next is our antigen-presenting cell travels to this tissue. And what happens here is the B cell itself is sensitized, enlarges, and divides. 3 really important terms that I've just highlighted that you should be including as you're describing this process. And as this B cell is sensitized, enlarged, and divides, we're obviously going to get clones of that particular B cell forming. Now, there are 2 options from here. Number 1, most of those B cells are then going to become plasma cells, and the plasma cells themselves secrete antibodies. Now, as we've mentioned previously, these antibodies combine with antigens to form that antigen-antibody complex. The second option that can take place here is that some of those B cells will eventually develop into memory cells, so that upon reintroduction of a former non-self cell into the body can remember and knows how to proceed with protecting you at the 3rd line of defense stage. So this here is a quick list, a checkpoint for you to tick off these stages involved in that humoral response. So as we've just drawn on our diagram, our first step is where the antigen-presenting cell migrates towards that lymph tissue. Secondly, within the lymph tissue, we've got lots of different types of B cells, and these are sensitized, enlarged, and caused to divide. Now, another word that we can use to describe this process is the proliferation of B cells into clone cells. Now, once we've had the proliferation of clone cells, we've got an optional extra step, and this will tie in more in our next lesson when we're talking about the cell-mediated response. But some of the B cells do require helper T cells in order to become sensitized. And we'll come back to this in the following lesson. But our main process here, as we've described, is that either the clone cells will become plasma cells, or secondarily, they'll become memory cells. Now, the key distinction here, these plasma cells secrete antibodies, but they're generally short-lasting. Memory cells, on the other hand, are very long-lasting, and should you get sick again, in what we will call from here on out a secondary response, the body can provide a really rapid response to that particular antigen. So here is another diagram representing this process. Again, I'm going to skip over it for now, but you might like to have a look through it, and one final representation here. So when we're talking about the action of the antibodies themselves, it's good to review these and make sure you've got notes for quite a few of them. We've got 2 main categories here. So the first sort of action of antibody is going to really enhance phagocytosis. So we're talking the agglutination of these foreign bodies. Secondarily, we can also have precipitation of soluble antigens, so preventing them from dissolving within a particular liquid. And finally, they can also coat bacteria and make them easier for other cells to ingest. From here, we've also got antibodies will bind to the surface of antigens, and what that's largely going to do is prevent them from entering cells and effectively neutralizing them to a certain extent. And then we also have a few options involved in cell lysis, so cell death. So we can use antibodies, and some might inhibit reactions or inactivate antigens, and that's going to cause cell lysis. And secondarily, some antibodies can also have the effect of dissolving organisms or causing a hole to form within the cell membrane, making it become more permeable. So that's our very quick overview of the humoral response, and following this one, it's good to have a look at the cell-mediated response.